Great. Welcome, Dr. Fadi. Welcome, Dr. Karin. Hi, everyone. Welcome to this very special discussion. Today, we are diving into one of the most exciting and complex frontier in transplant medicine, regulatory T cells. We're incredibly lucky to be joined by two leading voices in this space, Dr. Fadi Issa, the chair of transplantation at University of Oxford. He also serves as editor-in-chief of BMJ Immunology and leads a groundbreaking and use of is to redefine transplant success. Karin, a physician scientist, life sciences executive, and founder of Lyfebulb, having herself navigated the journey of living donor kidney and pancreas transplant, she combines clinical expertise from the Karolinska Institute, along with a deep personal commitment for improving the life of transplant recipients. Dr. Issa, Dr. Karin, thank you so much for joining today for this conversation for the benefit of our patient community. With that, I'll step back and hand over the floor to Dr. Karin. Dr. Karin, over to you.
Thank you so much, Abhishek, and it's really an honor to be here. Thank you so much for giving us the opportunity to talk about this very interesting topic. And I'm really honored to be together with you, Dr. Issa, because you are truly a legend in this space, and it's really nice to share the floor with you. And I know I'll learn so much during this time. So just a little bit to set the stage. As Abhishek mentioned, I'm a fellow patient. I have gone through 2 kidney transplants, 2 living donor kidney transplants, and 1 pancreas transplant. So I've gone through the process 3 times, and I'm managed now with chronic immune suppressive therapy. And I think importantly, the reason for my 2nd kidney transplant was mainly due to fibrosis of the first, meaning my father's kidney. And it was very, very difficult to realize that although I had tried to maintain, a very thorough, adherence protocol and do what a patient should be doing as a transplant patient, my kidney still failed. And it was really mostly due to the medication that I was on, the very medication that is supposed to protect my kidney from rejection, which is why you use immune suppressive medication, because the organ that you receive is not your own, and therefore your body's immune system is primed to try to reject. Even over time, it doesn't get better. You still have to manage your immune system. And so I've been really dedicated to this space, not just for my own, condition, but also because of all the other patients who are going through similar situations. And my primary objectives have been to increase the number of transplants every year so that we can serve many more patients who are in end-stage disease, kidney disease, but also liver and heart and so on. And but also to help manage those who do receive transplants better, so that they have a better quality of life and a better, longer graft life. So one of the interesting areas, of course, is better drug development, meaning new drugs into the space. And most of the time that is looking at small variations of what we have right now. But the reality is that the transplant space hasn't seen a new drug into the market since 2011, when we had belatacept enter the market. And we are in desperate need for something better than the current regimen. So I'd love to ask, Dr. Issa, how do you see, first of all, the current landscape of what is available for physicians to provide to patients who have gone through kidney transplant? How good do you think that is, the portfolio of opportunities? And then we can go to what's coming in the future, and in particular T-REGs.
Thank you very much, Karin, and thank you very much for having me, and thanks very much for the very kind introduction. I mean, I'm an expert in this narrow area of the field, but no one is as much an expert as a patient. And I'm always hugely grateful to speak to patients and understand their experiences, and to have someone like you, who is a patient and an advocate for the field, trying to push things forward, really that's the best that we can have. So thank you very much. You are absolutely right in that the field of transplantation actually has not had big improvements over the past few decades. And there is this sort of myth publicly that transplantation is solved. People have kidney failure, they have a transplant, and everything's okay. So it's almost like a medical success story, and everyone thinks it's not really a big issue. In fact, when you speak to the public, the thing they mostly worry about is organ donation. That message has got out that everyone should be an organ donor and we need more organs. But actually, the fact that transplantation is not a complete success, that there are still big challenges there in terms of how to manage patients post-transplantation, I think that message has not got out. And I think to a degree, the field has done itself a disservice by saying what a big success it is, and actually not saying that we still have these major problems to solve. And they are major problems, as you very eloquently told us. Patients have a limited lifespan in terms of their transplanted organ, because the transplanted organ is subject to the immunosuppression, and the immunosuppression is directly toxic to the organ. It's crazy. The drugs we give to stop rejection are actually directly toxic to the organ. And so that is a problem. And the immunosuppression now is actually the cause of the greatest morbidity, the greatest side effects for patients, rather than rejection itself. So what do we have now in our toolbox for patients? Again, the reality is the drugs have not changed a huge amount over the past few decades. We're still using similar drugs that we used in the 20th century when transplantation began, when the inception of the field. So that is a problem. We are targeting the same cellular processes, the same cellular mechanisms to try and suppress immune responses. And we have yet to find something which is specific against the transplant, against the rejection response against the transplant. Whereas most diseases you think of now, they're drugs that really target only the pathology, drugs that only target this. You have an inhaler, for example, for asthma, to dilate your bronchus. You don't have something that dilates every vessel in your body, right? Whereas here, what we're doing with immunosuppression is we are suppressing all T cells, every single T cell, every single immune cell. It seems completely unnecessary. We have had belatacept come on board, and that is a big advance, but it is still non-specific. It's directed against all T cells. So again, it doesn't have the specificity that you want against the transplant. So that is where the field has not made any major advances, I would say. And we are still using the same tools we had from many years ago. Most patients take tacrolimus and mycophenolate mofetil. That's pretty standard of care, and steroids. Some are then they may be transferred onto things like mTOR inhibitors, so like sirolimus or everolimus, belatacept, but really not a big change. And why is that the case? Well, partly is because cynically, I would say the drug companies don't necessarily see a good reason to invest, because the drugs that they do have are hugely effective at short-term survival. So if you measure things with short-term survival in terms of 1 year patient survival, 1 year graft survival, actually they do very well. So there's no real incentive for them to look at 10-year, 20-year survival. So that's my cynical take on things, but I would hope that there's some hope coming, and there are things that we're working on.
Yeah, maybe I really appreciate that, and I think that is exactly right. I think the other problem is the regulatory pathways, right? Because for the drug companies to want to invest in a new approach, they need to know that they can get approved, through the FDA or the European authorities, to be able to launch their drug, because there's a lot of risk and a lot of money involved in developing a drug. So if the endpoints are looking at this 1-year graft survival, where we already have 95% success, there's nothing you can do to be better.
Yes.
And even these non-inferiority trials are just not very attractive from a so-called marketing perspective or market positioning. So we need to change the view of the regulatory authorities, as well as the people who are financing the drug companies, that real life with a transplant is not good enough for patients and we can do better. So I think it's up to scientists like yourselves to really show what this could look like for a patient going forward. And with that, I'd love to hear more about what you're working on and this field of T regulatory cells. And first of all, what are T regulatory cells?
Yeah, thanks. Yeah, so T regulatory cells are a cell therapy. So we all have these. It's a subtype of T cells. T cells are a type of white blood cell that are part of what we call the adaptive immune system. So they're there to fight bugs, so viruses, bacteria, but also they're there to detect cancer if it develops and to kill cancer. And so you might have heard of these CAR T therapies in the news, and those are a particular type of T cell which has been programmed to kill a cancer. So they're there to kill usually blood cancers. We are flipping this and looking at regulatory T cells. Regulatory T cells do the opposite of killing. So they're there to suppress immune responses. They're there to put a brake on immune responses. And we all have them, and they are invaluable because in people who have defects in regulatory T cells, they develop overwhelming autoimmune disease. So that tells you how critical they are to balancing the immune system. They're there to keep a check on all immune responses. So what we're looking at is taking those regulatory T cells and making them defensive against the transplant. That's the thinking behind it.
So they would be protecting the transplant?
Correct. Exactly. So I'll go into a bit more detail about what they are. So in terms of how you identify them, they have specific markers on the cell surface, which means that you can use antibody techniques to isolate them specifically. And you can take them from patients, and then what you can do is grow them, what we call ex vivo, expand them ex vivo in a dish or a bag, for example, and then return them to the patient at higher numbers. And the thinking is that rebalances the immune system so that it is less forceful in attacking the transplant. That's the whole idea behind it. And people are also working on ways to make them specific to the transplant. So you can put a receptor on them, which means they only suppress immune responses that are directed to the transplant. So that's the idea behind regulatory T cells. And you may have heard last year that they were also the subject of the Nobel Prize in Medicine and Physiology, the discovery of regulatory T cells. So clearly they are very important physiologically. They have important therapeutic effects, and people are also looking at using them for autoimmune disease, because if you can suppress immune responses against a transplant, you could potentially suppress immune responses that are autoimmune, and people are also investigating that as a cell therapy.
And how do you make sure that the organ that you have received, the transplanted organ, that you are making sure that it is fully represented by the T regulatory cells that you're expanding ex vivo?
It's a great question. So at the moment, there are two approaches. One is to use something called polyclonal T-REGs. That means these are T-REGs that I take from a patient, expand, and so they have receptors on them that recognize different antigens, recognize different foreign proteins. And so around 10 to 20% of those recognize the organ. In the same way that you have T cells that recognize the organ to reject it, you have T-REGs that recognize the organ to protect it. So it's the same percentage of cells, 10 to 20%. So if you were to take these T-REGs, expand them so they sort of are higher than the number of T cells that reject the organ, then you suddenly have a proportion of T-REGs which recognize the organ and protect the organ. So that's one way of doing it. The other way is to say, I know the HLA type of the organ that has been transplanted. The HLA type tells you the foreign proteins that are on the surface of the cells of that organ. And then you create a T-REG with an artificial receptor against that HLA type. And that's called a CAR T-REG. So that's the other approach that you can take. Both have their merits, and both are being actively investigated.
If you think about it from a patient perspective, a protocol perspective, in the first case where you would just try to create more of the T-REG cells than the T cytotoxic cells or the cells that are killing, distribution, when would you expand the cells in the order of, if you think about a patient getting a kidney transplant?
Yeah, great question. So that's a bit of a live question. At the moment, what we are doing in kidney transplantation, we've done a phase 1 trial and we are just towards the end of a phase 2 trial now, is we have taken blood from patients pre-transplantation, isolated the T-REGs, and then expanded them, and then we have frozen them into cryovials that we can keep for when we need them. And what we have done is given them at 5 days post-transplantation, 5 days after the transplant. And the reason we've done that is because we think that's when most of the recognition of the foreign organ is happening. That's when immune responses are starting to build against the foreign organ. And the T cells that are recognizing the foreign organ are starting to also expand in the patient. So that's probably a good time to say, wait a minute, here are some T-REGs that are going to counter that effect. So that's one way of doing it. Another way of doing it is to give them quite late. And we've tried that as well. So to give them 6 months after transplantation. But that we've done in the context of patients who receive what we call lymphodepleting immunosuppression. So they're getting immunosuppression that completely wipes out all their T cells, all their lymphocytes, T cells and B cells, and it wipes out their T cells for about 3 to 6 months. So we think actually giving T-REGs in that situation is not necessary, and we're not sure what they would do in an environment which is lymphodepleted. So we give them at the point that we think T cells are starting to come back at 6 months. So those are the 2 points that we have tried. But I think perhaps there's an argument for also thinking about giving them during a rejection episode, for example, but that's something we haven't explored yet.
So the former is a more benign approach in the sense that the latter one is highly, I would say, invasive in the sense that for the patient needing a full depletion, meaning going through essentially chemotherapy. Would you do this after? When would that happen in the timeline of the patient?
Yeah, so the lymphodepleting therapy that we use actually is one that we've used in Oxford for a while. It's called Campath or alemtuzumab, and in fact that is standard of care in Oxford, believe it or not. So in Oxford, what we use is Campath for patients receiving kidney transplants. The reason we do that is because we have also had some trials where we've compared Campath with other induction immunosuppression, with basiliximab, and we found that Campath had better outcomes. So we wondered, how can we insert T-REGs into what we know is standard of care here in Oxford? But equally, we know that that's not the case in other units. And in fact, most other units don't use Campath. And so we thought, how can we actually give T-REG therapy in that context? And that's why we
do you use Campath for how long post-transplant?
It's just around the time of transplant. So usually just pre and post. exactly.
Yeah. Okay.
Yeah, yeah.
So it's a similar protocol to using thymoglobulin or
Right.
ATG in the United States. Okay.
Exactly. Yeah. Yeah.
And
So
basilic as
and
well.
Basilexab, yeah. So you can envisage a situation where patients have their standard of care ATG, anti-thymocyte globulin, and then have T-REGs later, 3-6 months, for example. Or you can envisage a situation where patients have T-REGs in the beginning at day 5 and no induction immunosuppression. So that was the benefit that we did there, which is that the T-REGs were actually replaced the induction. So those patients had no induction immunosuppression at all. So that reduces their immunosuppressive burden, from day 1, basically.
And in your phase 2 trial that you're in now, do you have a standard of care as tacrolimus and mycophenolate
Correct.
on top of the T-REGs, or
Right.
are they?
So at the moment, what we're doing is a randomized control trial. So patients are enrolled and then they get randomized to either Treg therapy or standard of care. Standard of care just have basiliximab induction, and then they continue on tacrolimus and MMF as per standard of care with no reduction in either. The Treg arm do not have basiliximab. They have Treg therapy on day 5. And then at month 6, we stop MMF.
And you do have MMF on top.
So they have MMF and tacrolimus. So T-REGs at day 5, no basiliximab. They continue on TAC and MMF, tacrolimus MMF. And then month 6, they stop MMF completely. And then they continue on tacrolimus monotherapy, which we then start to try to dial down safely to at low levels. We try and achieve low trough levels of tacrolimus. So those are the two. So the standard of care just have standard of care immunosuppression, tacrolimus and MMF, basiliximab induction, and they carry on as per normal with both. The T-REG arm, no induction immunosuppression, and they stop MMF, and we dial down the tacrolimus. So that's the current trial. In the phase 1, although we weren't actually planning to stop immunosuppression because it was a phase 1 feasibility safety trial, it was an option that was built into the trial protocol, and in a few patients we decided to try to take them off MMF and try to reduce their tacrolimus, and they all did very well. So none of those patients had any rejection episodes in the phase 1 trial where the patients had T-REG therapy.
And they have continued on now?
They have. Yeah. So that phase 1 trial is now, we have 10-year follow-up data from that. And the patients in the T-REG who received T-REG therapy, who have come off immunosuppression, have all done right. In fact, we have not had any rejection events in the T-REG therapy arm. And the other interesting thing that we found in that arm is that they had fewer infection events as well. So we found that overall they had lower levels of CMV, seroconversion, lower levels of EBV, and overall lower levels of bacterial infection as well. We think that's probably just due to reduced immunosuppressive burden. So because those patients didn't have induction immunosuppression, they didn't have basiliximab, and then some of them have come off MMF, I think overall they've got lower burden of immunosuppression. The other thing that we found is that they've had much better graft function over time. So their EGFR has been maintained high throughout over that 10-year period. And we compared them to patients who didn't have T-REGs, and unfortunately, there have been some graft losses there.
And your trough level for the patients who received T-REG therapy was around, what was the tacrolimus trough level that you maintained?
I mean, we're sort of aiming for around 4, and maybe sometimes slightly under 4 if we can.
Okay, that's good. That's probably why the graft function is better 10 years later too. Very interesting. Just so I understand more about the T-REG infusion in the patient, you have then taken T-REG cells and expanded them. So you took the T-REG cells from the patients prior to transplant and stored them. So how long before the transplant did that happen?
So what we do is we take a unit of blood from the patient. So it's just, standard blood donation, as it were.
Yeah.
And then we isolate, expand, and freeze them. And we aim just so, in terms of dosing, it might be helpful for people to know around 10 million cells per kilogram is how we do. So kilogram body weight of the patient. And then they're cryopreserved. And we have the ability to keep them frozen for actually many years. At the moment, the regulator says we can keep them for up to 4 years. So that process of making the T-REGs can happen well in advance of the transplantation. Can happen many months. So it can happen at the point at which patients are listed. And it takes about 6 weeks, 4 to 6 weeks to make the T-REG product and freeze it. So as long as you can do it within that window, you can then treat up to 4 years down the line.
4 years. Okay.
Yeah.
Are there any risks with having too many T-REG cells or expanded T-REG cells?
It's a great question. There are theoretical risks, certainly, in that you can envisage by giving more cells that suppress, well, perhaps you'll end up with more infections or more cancer developing, for example. Those are theoretical risks that, as it stands, have not been borne out in the data, in that the patients haven't had more infections, for example. In terms of malignancy development, looking at the phase 1 trial where we have 10-year data, because it's very difficult to look at cancer development until many years down the line, they've had an equal amount. So we haven't seen more or less in either group in terms of cancer development. So those are the potential risks. But I think what's important to note is that the T-REGs that we're giving back are the patient's own T-REGs. They have the T-cell receptors of the patient. And so they are there physiologically to stop autoimmune diseases from developing. So giving back that sort of type of T-REG back with those, I would say, clonotypes, those TCRs, doesn't seem like it would be, so again, theoretically, doesn't seem like it would be detrimental. But it is a risk. And that we worry about, that are we doing the same thing that immunosuppression does, but just giving them back as cells? But we haven't seen that happening. We have in animal models, so a lot of this obviously is lots of animal models to get to this point. Given animals Treg therapy and then infected them with a virus to see whether they can still mount a response against that virus, and they've been able to mount a normal effective response. So we haven't seen that effect in animal models. So I would say there is a theoretical risk, but we haven't seen that borne out in the data.
And you also mentioned autoimmune disease. Many patients who reach end-stage kidney disease or kidney failure do suffer from some sort of autoimmune disease, lupus, type 1 diabetes. Do you see any effect from the extra number of T-REGs on their original etiology?
I mean, it would be very interesting to know whether it's protective against those pathologies as well. Treg therapies are being tested actively in diabetes. So there have been some trials giving T-REGs to children with newly diagnosed diabetes to see whether you can prevent onset, reduce insulin requirements. And some of those trials are actually very promising, particularly some work from Poland, from Piotr Trzaskowski, where giving T-REGs to children seems to perhaps have a positive impact on their C-peptide levels. So that's health of their pancreases. So that sounds very promising. And there's some work in California as well from Jeff Bluestone looking at using T-REGs there. And people are now turning towards perhaps using T-REGs for rheumatoid arthritis. And there are some companies in the US who are starting to look at that. Sonoma Therapeutics and also Quell Therapeutics in the UK are now looking at using T-REGs for prevention of rheumatoid arthritis, another autoimmune disease. So there's definitely interest. I would love to know whether the patients who've received T-REGs for their transplants have reduced autoimmune disease development, but I think we just don't have the power in terms of the number of patients to be able to detect that effect.
Yeah. No, it's interesting because you're essentially giving a very wide range of T-REGs. It's
Correct.
Not specific to the. You mentioned there were two things you could do. One was to just increase the number, and the other one is to be more specific to the actual HLA type of the organ you receive, which would make it possible maybe in the living donation, but probably not for a disease donation. But yeah.
Tricky for deceased donation, exactly, because you don't know who the donor is. I mean, one thing that we're working on at the moment, but it's still in preclinical testing, is to develop allogeneic regulatory T cells, so not having to make them from the patient, but to make them in advance from a healthy donor and bank them for when you need them. And in that situation, what you could do is you could then program them across multiple different HLA types so that you cover the population. And people have calculated that you need a certain number of HLA reactive CARs to cover most donors. So you would have an A2 and an A3 and an A4, etc., so that you can cover most. And if you have that bank of T-REGs made from a healthy donor, you could then use the one that you need in the event of a deceased donation, where you know on the day this is the HLA type, you take those T-REGs and use them. That could be the future.
And that's a much more commercially attractive play, where you could have off-the-shelf T cells.
Correct.
Versus having to go through the ex-vivo expansion and
correct. So you can do it all in advance. Exactly.
Yeah. There's a company that does that. I mean, several, but in the cancer space, Sana, right? Sana Biosciences.
Exactly. So they're working on that, and they've had interesting success, I believe, in islet transplantation as well. Right. So Sonia Schrepfer has had that success with reducing HLA expression on islets, and a very interesting case report of a patient who received islets and became insulin-free.
Per-Ola Carlsson in Uppsala.
Exactly.
I'm very familiar with it. I think it's really fascinating, the hypoimmune platform in the cadaveric islets, and hopefully in the future moving into more of the islets, the expanded islets.
Absolutely, absolutely. So, I think that's a really promising area. So what we've done is actually quite similar. So we've reduced HLA class 1 and 2 expression on T-REGs so that they become hypoimmunogenic, and then we've shown that in preclinical models that they work. So it's now starting to think about how we might translate that therapy towards a clinical production process.
Interesting. So that brings me to a question on how you're doing this in the academic. I don't know, the Google Meet keeps moving me back and forth. I'm not used to this technology. In Zoom or Teams, you're kind of staying,
you're
stuck.
I'm
not moving, by the way. It's just anyway. It's funny. Are you doing this in the academic setting of Oxford? But are you in a phase 2 trial? Is that a company-led trial, or is it an investigator-initiated trial?
It's investigator-initiated, so this is all completely academically led. And I think there are benefits to both approaches, to going investigator-led or not. I think one of the challenges in general with cell therapy is often there's no intellectual property to protect, that companies will think, oh, I'll take that and make it. What CAR T-REGs? Yes, because there's the CAR receptor which you can protect, and that's why that got commercialized. But that doesn't necessarily mean that's the better therapy. I think it was just commercializable. That's the point. So we've gone down this route of keeping everything academic for now. And my view on how to progress this type of therapy is, at the moment, to try and build infrastructure within healthcare where you can produce cell therapies, so that actually it can be done within hospitals when they need to do it. Now, there's a lot of work that needs to be done there in terms of making the process for production easier and closed, and something that you can distribute in different hospitals. But that's the route that I think holds some promise, and you can compare it to, for example, stem cell transplantation, which is not commercial. It's done usually in hospitals. Healthcare providers isolate stem cells for bone marrow transplantation, give them back to patients, and they have the ability to do that. At least we do in the UK in NHS hospitals. And we also have in the NHS at the moment cell therapy production units, which are slowly building up. So my sense is that it would make sense for this type of therapy to just enter healthcare and be produced in that way. It would be cost effective, and not dependent on having to be commercialized.
Do you have an assumption or an idea of say in the United States? I mean, or if we stick to Europe, but if you have a speculation on the United States, we start in the UK. Of all your transplant centers, how many could be doing this kind of approach? What percentage?
Yeah, I mean, so I don't see why they couldn't all do this approach, is the reality. What you would need is to have a T-REG production process distributed geographically. So we have at the moment NHSBT cell therapy production units, which are across the country in London, Oxford, Bristol, etc. So you could have them make the T-REG therapy and then distribute it to the transplant centers outside, which is essentially what we do at the moment. So the cell therapy is made in London and it's transported to Oxford when we need it. So you could envisage the same thing happening. I think it would be very difficult for one single center to make it for the whole country logistically, but you could have multiple centers.
You could have hubs,
exactly,
centers of excellence.
Exactly. Yeah.
And then just distribute, and it would be the same the size of the country, the United States, very large, but you could have a West Coast, East Coast, Central, and so on. Very interesting. Yeah.
I mean, you have the organ procurement organizations who are distributed, aren't they? And you can envisage a similar kind of, maybe they link into where they are geographically, because that also makes sense that perhaps it goes with.
You are still pursuing a clinical, a clinical trial in phase 2 and then eventually phase 3, where you would need a large, and your goal is to get a label for this, or what is the goal or prove that it works? Since it's not a proprietary product, what are we hoping to achieve with the clinical trial?
Yeah, so I mean, so it's an interesting question, and it's not necessarily clear whether we definitely do need a phase 3 trial or not. So I think things are changing. There's a lot of change in terms of the required level of evidence, and sometimes phase 2 trials give you the evidence that you need to then go to the regulator and say, this is no longer produced under clinical trials or under a specials license. This is now, here's the production, here's the manual for production, this is how it's made, these are the QC, the quality control attributes of the product that we expect it to be made to, and these are the release criteria. And then you would then go to what we have. So in the UK, we have NICE, which is the National Institute for Clinical Excellence, who then assess new therapies and say, yeah, this is actually something that makes sense economically, and has benefits, and they assess things like QALYs and so on, quality associated life years, and then say, yes, this is a therapy that we should commission in the NHS, and so then the NHS commission it as a therapeutic, basically.
Yeah,
that's the hope. That's the hope. We'll see.
That makes sense.
Yeah.
Well, I wish the best. Do you need patients to be? I mean, how do you recruit patients to this? Has that been difficult, or how?
Yeah, it's in fact, patients are the easiest. I mean, they're so keen. They're always. I'm always just amazed and humbled by patients and what they're willing to take on in terms of additional burden of a study and the risks of a study. But I think we've maybe had one, perhaps two patients decline to go into the trial. We've actually completed recruitment as of this month, which is great. So we're now, thank you very much. So we're now into the long-term follow-up. So hopefully we'll have some useful data in a year or so once the last patient.
That was what I was going to get to next. When can we see the data? Next year?
Fingers crossed in about a year.
So that would perhaps coincide with the WTC, or would when do you think you would present it? Yeah.
Yes, it might do if we're lucky. As long as we can analyze and clean up the data in time. I've always wanted to try sort of find out what's going on, but I'm blinded to the results, and I'm going to maintain that for now until we actually have the results ready.
But sometime during 2027, you think that you will be able to release the data?
I hope so.
Well, that's quite exciting. Abhishek, anything that you would like to ask Dr. Issa?
Yes, definitely. I had two questions in mind. Thank you for all the knowledge you've shared with us, Dr. Fadi. I'm sure the patient community are, they'll be holding their breath for 2027 results to come out. I had two questions which I wanted to touch upon. First was regarding the commercial aspect, which you already gave us a little bit of idea that there were companies like Sangamo and Quell who had they met their kind of clinical objectives and their trial results for their phase 1, phase 2, but then decided not to invest further down the line, and they switched to autoimmune diseases as a lead problem statement to solve. I just would hear your thoughts that do you see this crossing that barrier of commercialization post successful results? That's my first question. And the second question, and you can answer both of them together, is you also mentioned CAR T therapies, which are now widely used. In terms of manufacturing process, how similar is the manufacturing process for CAR T therapies versus what is required for T-REG cell?
Sure.
And if cost is sort of the consideration, and if the process is similar, could companies who already have GMP facilities for CAR can they kind of, with some modification, reuse for T-REG down the line?
The
reason I'm asking, they are like ImmunoAct, who recently have successfully brought down the cost of CAR therapies by a factor of 5 to 10, and are working exactly the same model where there are certain center of production who work with hundreds of hospitals around the country in India for delivering a much lower cost CAR T therapies. So these were two questions I had in mind. So if you could
great questions. I mean, my view is we cannot depend on industry to save us here and make these cell therapies for us. We have to do it ourselves. You've exactly explained the problem. They have pivoted to where there's more patients, more money, autoimmune disease, where they have the ability to make more of an impact in that way. And transplantation, where there is a need, but to them, it's actually a narrow population of patients who, again, to them are doing quite well. they don't. It's actually very difficult for them to put. So this, I believe these are all financial decisions, is what I'm saying. I don't think these are scientific decisions. This is a financial decision where they have a proof of concept in transplantation. They've shown that they can make a CAR. They've the feasibility, the safety. They can do it in transplantation. It works. Tick. Now let's pivot to where there are more patients who could be treated. That's the reality of what happens there. And actually, that is the reality in general of the development of drugs to target the immune system, is that most of them are focused on autoimmune disease or cancer, and not transplantation. And those are market factors and financial factors. I don't believe they're scientific factors. So that's my view on what's happening there. In terms of, can a cell production suite make CAR Ts? If they can make CAR Ts, can they make T-REGs? I don't see why not. Yeah, I mean, I think they're very similar processes. There's isolation of cells, it's expansion of cells. I mean, the isolation is different in that you need to isolate a different population, but that's fine. It's just a change of process. And expansion, again, slightly different because they need different nutrients. But again, it can be done. I don't see that necessarily as a barrier. I would say that T-REGs, for some reason, are a bit more difficult to make than T cells. T cells are always very happy to expand. T-REGs are a bit more picky, and in fact, what we worry about is a small population of T cells in the T-REG product because they expand a lot and they overshadow the T-REG population. So it is slightly trickier, but I don't see why it wouldn't be possible to make them.
Understood. So let's say if we are able to manage the cost factor, then you see this could be a widely adopted model where hospitals themselves are producing it, and or let's say lead hospitals are themselves producing it in various parts of the countries, and then those T-REG cells are like shipped across different centers where they can be consumed. That's a viable model.
That's what I would hope. Yeah, exactly. That's what I would hope. I mean, we have done some health economic analysis and shown that if T-REGs are successful at reducing a certain percentage of rejection events and graft losses, that actually the balance of the cost of the T-REGs is balanced out by reduced complications, reduced retransplantation, etc., etc. So I think financially it could be a viable thing to do, and as more and more people make it, and as the process becomes simpler and simpler, costs come down. I think that's the reality. I mean, we make T-REGs at 1/10 of the cost of what industry makes CAR Ts, right? So that's academic. We can do that, and similarly, I think healthcare providers should be able to do that as well.
I mean, in the U.S., we've seen, this movement toward making islet cells a product, and that has been very contested by academic institutions, including leading surgeon Piotr Witkowski, right, who is doing a great job now, but keeping patients off tacrolimus and using Tegoprubart. And it's got a lot of attention through social media.
Absolutely.
I think entire Instagram is flooded with reels about this particular trial. And I think we need to do similar thing about T-REG therapies for transplant as well to get the right attention.
I mention it because I think it is a similar path in the sense that I had not heard. I mean, I really am grateful for this opportunity to learn more, and I think we need to really make more noise about what's going on, because you do have some great early results. It'd be nice to see the phase 2 next year, and that's when we really have to think about how to reach the patient community and also how to set this up in more places.
Absolutely.
I appreciate this.
That's the dream. Yeah. Thank you very much, Karin.
Thank you for what you're doing and taking this risk here for all the patients who could benefit. So I really appreciate that.
It is. It's for them in the end. Absolutely.
Yeah. Right.
Yeah, I think we can wrap that up with those thoughts. Thank you so much, Dr. Issa and Dr. Karin, for like on behalf of entire patient community. I think we all are today, and all of us are kind of keeping our fingers crossed for results we publish next year, and we hope to keep hearing from you down the line as well. Thank you so much for all the work you're doing.
Thank
you
you,
so
thank
much.
you both
Thank
for
you.
thank you both for the opportunity to tell you about what we do, and yeah, and hopefully this reaches some patients, and I'm
Absolutely.
sure I'll see you soon.
Thank you.
Thank
Bye.
you. All the best. Bye.